Okay, now initially, scientists, the idea of the biochemical reactions always very restricted last time, because we don't have a sufficient, in advance in terms of the technology to know about the protein and enzyme. So scientists thought that enzyme is everything, because without the enzyme, biochemical reaction cannot take place. So scientists actually classified enzyme as one category, not part of protein. So it come out, the first thing here is why cells know what to do is because of the enzyme. So they thought that DNA, the gene, actually code for the enzyme only. Gene give the instruction. Oh, now we need to carry out in this case, after the lunchtime, we have the high carbohydrates, so we need to convert, glucose sugar is very high, we want to convert the sucrose, glucose into the glycogen, insulin release. Are you clear? So we have the enzyme. Enzyme do everything. Okay, so scientists thought that first, enzyme that catalyze specific chemical reactions in the cells. So therefore, we know that gene dictates the phenotype through the enzyme. So it come out the first hypothesis known as one gene, one enzyme hypothesis. Okay, this is the first version, one gene, one enzyme. And after that, scientists realized that, not correct. Why? Because we still have other protein. For example, receptor, hormone, they are not enzyme. Can you see that? Are you clear? Hormone, transport protein, glycoproteins, they are not enzymes. So they scrap off this idea and change it. First of all, not all protein are enzyme. For example, we have the structural role enzyme. For example, we learn about collagen, we learn about keratin. They are non-enzyme. So they come out with, the revisions to this. Revisions to this one gene, one enzyme, and change it to one gene, one protein hypothesis. Okay, so they change it to one gene, one protein, because they know that, now gene also code for other type of protein where they are not the enzyme. Are you clear? Okay, so this theory or this hypothesis now is termed as one gene, one protein. But with the advancements in terms of the protein analysis, we do know that some quaternary protein, they need more than one gene. Are you clear? Okay, for example, hemoglobin. Hemoglobins, we do have two genes, involved. So can I say one gene, one polypeptide? No, in this case, you can't say one gene, one polypeptide. So therefore, now the final versions of the hypothesis saying that, so one gene, one protein hypothesis revised again now into one gene, one polypeptide hypothesis. Okay, one gene, one polypeptide hypothesis. Okay, now this way of saying one gene, one polypeptide hypothesis, from the way it's said, it's a bit confusing when it comes to the eukaryotic cell. So what is the problem of the eukaryotic cells? Because eukaryotic cells, one gene can code more than one polypeptide. Are you clear? In eukaryotic cells, one gene can code more than one polypeptide. But in opposite way we talk about it, one polypeptide definitely coded by one gene. You get the idea? One gene can code different type of polypeptide. Polypeptide 1, polypeptide 2, polypeptide 3, polypeptide 4. But when we talk about one polypeptide, it must come from one gene only. So that's why the way we say here, one gene, one polypeptide, it means that one gene code for one polypeptide, which is not true for eukaryotic cells. Are you clear? Okay, but if you talk about it, any one single polypeptide, it must be coded by only one gene. Are you clear? Okay, one polypeptide must be coded by one gene only. Now, you learn about this antibody. Have you learned about structural antibody? You have the antigen binding sites, correct? In the antibody, right? So we have so much variations of the antigen. We have so much variety of species of the pathogen. So in order to form this antigen binding site, we need to do a randomization onto our gene. So they create different shape, correct or not? Each type of shape is one polypeptide. Each type of shape is coded by one polypeptide, one gene. Each one polypeptide coded by one gene. So in this kind of randomization process, actually, for one gene, you can create many kinds of the polypeptide. And you see that particularly involved in the immune system. Okay? So this we call randomization, where we're going to learn it later when we talk about the transcriptions and the post-transcriptional modification. Okay? So with this, I've done for this one gene, one polypeptide hypothesis. It's more to the theory and very factual. Right. And for the recording now.